everyone, and welcome to this new episode of Ask Stago, the podcast from Stago. This platform is dedicated to answer the questions that you may have about our products or hemostasis in general. My name is Lisa Gannon, and I'm Global Product Manager here at Stago, and I'm so glad to join you here today. To animate today's session, I'm joined by Anne-Cécile Ortega, also Product Line Manager at Stago for Lab Automation Solutions. Hello, Anne-Cécile.
Hello, Lisa. Hello, everyone.
Today, we're going to discuss a very specialised topic, the need for better standardisation of anti-10A reagents for heparin monitoring.
Yes, Lisa. And to cover the topic with us, we are glad to welcome Dr. Gouin-Thibault. Dr. Gouin, it's a pleasure to have you with us. You joined us from the University Hospital of Rennes in France, where you are medical pathologist and the head of the Hemostasis department. You're also affiliated with INSERM and the University of Rennes, where you focus on research in hematology and hemostasis. Your research interests include antithrombotic agents, the variability of response to these drugs in specific population, and the role of antiplatelet drugs in modulating thromboinflammation. Dr. Gouin-Thibault, you're involved in several professional societies. You're co-chair of the Scientific and Standardization Committee on Control of Anticoagulation of the ISTH. You're the coordinator of the Thrombosis, Anticoagulants, and Antiplatelets group of the French Society of Thrombosis and Hemostasis. You're a member of the Peripathic Homeostasis Interest Group, and you're co-president of the Scientific Advisory Board and member of the Steering Committee of the Investigation Network on Venous Thromboembolism. Your ongoing contribution to the fields of hemostasis and thrombosis makes you a leading expert and a key figure in advancing research and clinical practice in these areas, which is why we are thrilled to have you with us today. So, hello, Dr. Gouin.
So, thank you very much, and good morning to everyone.
So, Dr. Gouin-Thibault, can we start with the basics? Could you provide an overview as to why unfractionated heparin is crucial in critically ill patients and also those undergoing cardiac surgery? And indeed, could you detail for us what the main challenges faced by the lab to ensure effective monitoring?
Well, as you all know, unfractionated heparin is the oldest anticoagulant that was discovered more than a century ago, and today, due to its short half-life and rapid reversibility, UFH remains the anticoagulant of choice in many critical ill patients. However, despite decades of use, the unfractionated heparin management is highly challenging, especially in these patients, mostly due to complex pharmacokinetics with chain of variable lengths and highly negatively charged. And one main issue with UFH is its non-antithrombin binding site. And it's very important to understand that when you administer UFH to a patient, the long chains will first bind to endothelial cells, plasma proteins, especially in inflammatory states, such as in a patient in ICU and also in cardiac surgery. And this results in heparin neutralization and high inter and intra-individual variability in response to UFH. And that's why it's very important to use a bolus. And also that's why monitoring of UFH is required to assess anticoagulant response and for dose adjustment.
So, Dr. Gouin-Thibault, anti-10A assays are commonly used to monitor UFH, but there is a significant variability in the result depending on the reagent used. Could you explain some of the factors that contribute to this variability?
Well, actually, unfortunately, so far there is no global test available to monitor UFH. So monitoring relies mainly on APTT or anti-10A assays, which are performed in platelet-poor plasma, except actually in cardiac surgery, where a CT is commonly used, because in this specific setting, we use very high concentration of UFH. And since APTT is not specific of UFH and is highly sensitive to conditions unrelated to anticoagulant effect, which makes its interpretation very difficult, especially in inflammatory patients, anti-10A is preferred in these patients. But the anti-10A assays, lack of standardization, and limited agreements between anti-10A values measured using different anti-10A reagents have been reported with a potential impact on patient management. And while one substantial difference between anti-10A assays is the presence or the absence of dextran sulfate in the reagent. And dextran sulfate is used in the first place to displace UFH from heparin neutralizing proteins, which are released in vitro by platelets after blood sampling, and so dextran sulfate is used to recover all UFH.
And indeed, one of your studies have highlighted that reagents containing dextran sulfate can significantly overestimate anti-10A levels. Could you elaborate a little bit on that and indeed the mechanism by which dextran sulfate can affect these measurements?
Well, as you said, we and others have shown in both in vitro studies as well as studies with quality controls, as well as studies with samples from patients, that dextran would, on top of displacing UFH from PF4 released in after blood collection, dextran could also displace UFH from complexes from in vivo with various protein, or also UFH complexes with protamine after UFH neutralization in the specific case of cardiac surgery. And such dissociations result in higher anti-10A values and likely overestimation of the actual active UFH in patients.
And how might the overestimation of anti-10A levels due to the dextran sulfate impact the clinical decision-making, and particularly regarding the management of UFH doses or protamine administration?
Well, it's been reported in different studies as the magnitude, and different studies and different settings also, that the magnitude of differences between anti-10A values measured using reagent with or without dextran could lead to changes in UFH dosage in up to almost 50% of patients. And in the specific context of cardiac surgery, in the Multicenter French study we conducted, after protamine neutralization of UFH, 77% of values were below the lower limit of quantification, which is expected when these anti-10A values were measured with a reagent not containing dextran sulfate. And when we use the reagent containing dextran sulfate, anti-10A values were greatly overestimated, with only 6% of value below the limit of quantification, which means that in this patient, protamine could be re-administered in a substantial proportion of patients with potentially deleterious effect.
Based on those findings, do you believe that there should be a move towards the standardization of the use of anti-10A assays to indeed improve the reliability?
Well, I definitely believe that we need a clear improvement improve standardization of anti-10A reagents, and also validation of these assays in different settings, but for sure.
And what further research or clinical studies do you think are needed to optimize UFH monitoring protocols?
Well, such studies are not easy to perform, but I really think we need additional data on patients currently treated with UFH, because these patients have changed from the first studies that were performed at the end of the last century. And so we need data with these patients, and we also need data with the reagents that are currently available, because once again, the first study were performed with reagents which are not used anymore. And we need to perform studies using these reagents, but along also with follow-up of clinical outcomes to confirm or redefine therapeutic ranges depending on the indications. And we also need robust data concerning the issue of altered response to UFH. And while, as I said, such studies are not easy to perform, so these issues could be addressed in future clinical research, maybe under the aegis of the subcommittees of control of anticoagulation and the perioperative and critical care thrombosis and hemostasis subcommittee of the ISTH. And meanwhile, within both subcommittees, we are currently developing guidance on the management of UFH that will be hopefully released soon by the subcommittees and presented at the next ISTH meeting. This guidance aims to offer standardized approaches and practical support for clinicians, including recommendations for the optimal methods for heparin monitoring, including assays and reagents to be used, with a specific focus on the presence of dextran sulfate in reagents.
And I'm sure that will be a very welcome addition. So thank you so much for this very informative conversation about UFH monitoring. I think we can all say we've learned a lot about the management of anticoagulation in this short podcast. And thank you to our listeners. As usual, all literature sources and links to previous podcasts are listed in the podcast description. But please feel free to send any questions that you may have to our email address, ask@stago.com, and we'll be really happy to address any questions in our following episodes. See you next time.
This podcast is brought to you by Stago. Diagnostics is in our blood.